Hello friends, I am Shruti Parashar. Welcome to my channel Biology 360. Today I am going to discuss the topic Haemodialysis and Peritoneal Dialysis. This topic is important for students studying in ICSE, CBSE, IGCSE and HSC board, and also for students preparing for various competitive exams like NEET, Olympiad and AIIMS. Hemodialysis. When renal or kidney doesn't function properly, there is accumulation of harmful substances in the blood. As we all know, the main function of kidneys is to eliminate harmful nitrogenous waste products. So, if the kidney is not able to function properly, there will be accumulation of harmful substances in the blood. In such a condition, the person has to go for artificial means of filtration of blood. In hemodialysis, blood is filtered outside the body using dialysis unit. So, hemodialysis is an artificial means of blood filtration. In this procedure, patient's blood is removed, generally from radial artery. It is passed through a cellophane tube that acts as a semi-permeable membrane. Cellophane tube is a thin, transparent sheet made of regenerated cellulose. And this cellophane tube, it acts as a semi-permeable membrane that helps in blood filtration. In this slide, we see the method of hemodialysis and peritoneal dialysis. In hemodialysis, one needle withdraws the blood containing waste products for cleansing through the dialyser. This blood is then carried to dialyser. In the dialyser, the blood gets filtered. Dialysate is also called as dialysis fluid or dialysis solution. It's a solution of pure water, electrolytes and salts such as bicarbonate and sodium. The purpose of dialysate is to pull toxins from the blood into the dialysate. The way this works is through a process called diffusion. And once the blood is filtered, it is returned back. So the other needle returns the clean blood from the dialyser to the body. In the other diagram, we see the method of peritoneal dialysis. In peritoneal dialysis, a small soft tube called catheter is surgically placed through the abdominal wall into the peritoneal cavity. Into the peritoneal cavity, dialysis fluid is poured via catheter. Then the blood is filtered with the help of dialysis solution, and then the peritoneal membrane, this is the abdominal cavity which lines, and it filters the waste and fluids from the blood, and the pure blood or the filtered blood is then again introduced into the body. The tube is immersed in a fluid called dialysate. Dialysate is isoosmotic to normal blood plasma. Only excess salts, if present in plasma, pass through the cellophane tube into dialysate. Waste substances move from blood into the dialysing fluid. Blood filtered is returned to the vein. Anticoagulant like heparin is added to the blood while it passing through the tube, and before resending it into the circulation, adequate amount of antiheparin is mixed. So heparin is a anticoagulant. It's a natural anticoagulant. Peritoneal dialysis. In this method, the dialysing fluid is introduced in abdominal cavity or peritoneal cavity. The peritoneal membrane acts as semi-permeable dialysing membrane. Toxic wastes and extra solutes pass into the fluid. This fluid is drained out after a prescribed period of time. Always remember, dialysis is regarded as a holding measure until a renal transplant can be performed, or sometimes as the only supportive measure in those for whom a transplant would be inappropriate. If a person is undergoing kidney transplant, immunosuppressants are administered. Immunosuppressants are drugs or medicines that lower the body's ability to reject a transplanted organ. Friends, subscribe to my channel Biology 360 and don't forget to click on the bell icon to receive the notifications on the biology videos. Also, please post your comments. I'm waiting for the comments. Thank you.